We know that angiosperms exhibit a wide range of diversity in their morphology, but their basic structure remains the same. Look at this angiosperm. This body consists of root, stem, leaves, flowers, and fruits. The morphology of any angiosperm plant consists of a root system and a shoot system. The roots, as you all are aware of, remain underground and develop from the radicle of the embryo. The shoot system remains above the ground and develops from the plumule of the embryo. The root system can further be classified into the taproot system that consists of a primary root and its branches and the fibrous root system. In fibrous root system, the primary root is short-lived and is replaced by numerous roots that originate from the base of the stem. All dicot plants are characterized by the presence of taproot system, whereas all monocot plants are characterized by the presence of fibrous root system.
You see this underground portion of the plant? Of course, they are roots. Roots help in absorbing water and minerals from the soil. But roots also have different regions that have specific functions. In this animation, we will learn about different regions of the roots. In this portion, you can see that the tip of the root is covered by the root cap that protects the delicate inner root tip. Slightly above the root cap lies the region of meristematic activity. This region consists of meristematic cells that are small, thin-walled with dense cytoplasm. Now these cells undergo repeated division, and that is why they are responsible for the apical growth of the roots. Now above this region of meristematic activity lies the region of elongation. The cells of this region elongate rapidly and cause the root to grow in length. This region is the region of maturation. Here, the cells from the elongation region mature and differentiate. Some cells of this region give rise to thin root hair that helps in absorbing water and minerals from the soil.
Let us study the regions of the root. The root is covered by root cap at the apex. Few millimeters above the root cap is the region of meristematic activity. Meristematic activity. In the region of meristematic activity, the cells are small, thin-walled, and they have a dense protoplasm and they are dividing repeatedly. After this, there is a region of rapid elongation, as you can see here, region of rapid elongation. In the region of rapid elongation, the cells of elongation zone are gradually differentiating and maturing, and this zone is proximal to the region of maturation. So this is the region of maturation. Region of maturation. In the region of maturation, from this region, the epidermal cells from the root hairs are there. So the root hair is present in the region of maturation. They absorb water and minerals from the soil, and from the root hairs, then this water and minerals through the xylem reach the stem, and from the stem to the leaves. So there are three regions of the root: the region of meristematic activity, the region of rapid elongation, and the region of maturation.
Have you heard of facilitated diffusion? I'll tell you what it is. In this process, a special carrier protein with a central channel acts as a selective corridor, which helps in the movement of molecules across the membrane. Now let us understand the process. Small uncharged molecules can easily diffuse across the cell membranes, as you can see on your screen. However, due to hydrophobic nature of the lipids that make up the cell membrane, water-soluble molecules and ions cannot cross the membrane. So they are helped across by transport proteins. Now the transport protein involved here is intrinsic. That is, it completely bridges the membrane. It also acts as a binding site for the specific molecule or ion to be transported. After binding the molecule, the protein changes shape and carry the molecule across the membrane where it is released. The protein then returns to its original shape and it waits for more molecules to transport. Small molecules such as oxygen and carbon dioxide move in and out of the cell in accordance to their concentration by diffusion. Let us now see how molecules are transported across the cell membrane against their concentration gradients. Active transport, you very well know, is the process by which molecules are transported in and out of the cell with the expenditure of energy. Let us take the example of sodium-potassium pumps operating in the cell to better understand this mechanism. Sodium-potassium ions are transported by special transmembrane protein channels. This protein in its original state has high affinity for sodium ions. 3 sodium ions attach themselves in this protein and an ATP provides the energy to drive this process. Now this binding of ATP causes the channel protein to change its shape and drive the sodium ions outside the cell. You can see that on your screen. Now, one phosphate group remains attached to the protein. The changed protein has high affinity for potassium ions and 2 potassium ions bind to the protein. Now what will happen because of this? This causes the phosphate group to detach from the protein channel and thus causing the channel protein to revert to its original shape. You see that on your screen? Now this conformational change causes the potassium ions to be released to the inside of the cell. Now the protein in its original shape is ready to take up sodium ions again. Now you would have observed that this transport of sodium-potassium ions is against their individual concentration gradients and it involves the expenditure of ATP. So this process is known as active transport. Smaller molecules such as oxygen and carbon dioxide move in and out of the cells through diffusion. The diffusion of water molecules from a region of higher concentration to a region of lower concentration across a semi-permeable membrane is known as osmosis. Let us take the case of solutes such as sugars, which are larger in size and therefore cannot cross the membrane. Sugars being polar will bind with water molecules. This binding causes a reduction of free water molecules outside the cell. This leads to the movement of water molecules from a region of their higher concentration to the region of their lower concentration. You must note here that with reference to the solute concentration, when the concentration of solutes is higher outside the cell membrane, then the solution is said to be hypertonic and the cytosol is said to be hypotonic. In this case, water moves out of the cell and this causes the cell to shrink. This condition is known as plasmolysis. When a cell is placed in a hypotonic solution, the water moves into the cell. The cell becomes turgid. When the concentration of solutes on both the sides of the membrane is equal, then the solution is said to be isotonic. In this case, there is no net movement of water molecules in and out of the cell.
Transpiration, as you very well know, is the loss of water from a plant in the form of water vapor. Water is absorbed by roots from the soil and transported as a liquid to the leaves via xylem. Now, in the leaves, small pores allow water to escape as a vapor and carbon dioxide to enter the leaf for photosynthesis. Right? Now, let us study the process of transpiration. In actively growing plants, water evaporates continuously from the mesophyll cells of the leaf and it creates a negative pull. The lost water in the mesophyll is replaced by the absorption of water from the adjacent cells. The xylem vessels of leaves pulls water from xylem of stem, which in turn takes water from xylem of roots. Thus, the water flow in a channel. Now, this water channel is held together by cohesion. Adhesion keeps the water column in place. When water moves into the xylem, more soil water is drawn into the root. This inward movement will continue until the soil becomes excessively dry.